Hello and welcome to the Clinical Care Options Infectious Disease Podcast. Today's episode is the second of a four-part series entitled An HIV Vaccine, An Unfulfilled Promise, which is produced in collaboration with the Global HIV Vaccine Enterprise at IAS, the International AIDS Society. This series features conversations between world-renowned guests in HIV vaccine development, HIV advocacy, and HIV policy to bust myths and address arguments for why an HIV vaccine is still needed and how we can continue working toward this ultimate game changer for durable control of the HIV epidemic. This episode features a discussion between Dr. Susan Buchbinder and Mr. Jontraye Davis. Dr. Buchbinder is the director of Bridge HIV, which is a prevention research unit housed in the San Francisco Department of Public Health and a clinical professor of medicine and epidemiology at UC San Francisco. Mr. Jontraye Davis is the community programs manager at FHI 360 and an adjunct instructor at North Carolina Central University. Their conversation focuses on what has been learned and created from 40 years of attempting to develop an HIV vaccine. For more information about Dr. Buchbinder and Mr. Davis, and for links to additional podcasts in this series, as well as HIV Unmuted, the IAS podcast series reflecting on the last 40 years of HIV research and advocacy, please visit the links in the show notes. Susan and Jontraye, thank you for joining me today for a conversation about what we've learned and built from decades of trying to develop an HIV vaccine. I asked Dr. Jerome Kim, the Director General of the International Vaccine Institute in Seoul, South Korea, to reflect on this question. Let's hear what he had to say to set the stage for our discussion today.
Yeah, so this is an important question. This idea that all vaccines are easy to develop or all vaccines are hard to develop is not going to be correct. HIV, the primary immune response does not control the infection. The virus mutates away from even broadly neutralizing antibody and away from cell-based immune responses. And so and then it inserts itself into the human genome. And so it goes into, permanent deep state. And so unlike COVID, developing a vaccine that will be safe and efficacious against HIV has not been a 12-month process. It's been a 40-year process. Are we closer now than we were before? Possibly. But there have been a lot of lessons learned. And so what are those lessons? So I think one of the first is that we learned a lot about the outside coat, the envelope of the virus. At first we thought it would be easy to make because we just come up with molecular biology and that became a vaccine, the vaccine from originally Genentech and then VaxGen, and it did not protect. So we've evolved along the pathway of trying to find the best immunogen. The other part of this is a simultaneous development of the broadly neutralizing antibodies. So the broadly neutralizing antibodies could potentially protect against lots of isolates, but we didn't know how to induce them. They were only found in 20% of individuals after about 3 years, and the immune responses weren't able to control the virus in the natural host. But what if you had them in the beginning? If your vaccine happened to induce them? And that started the next generation of vaccine development, and we're getting there now. And hopefully this new set of vaccines, maybe informed potentially by some of the work that was done with VRC01 that showed, in fact, VRC01 can protect against infection from VRC01 sensitive viruses. So again, we've learned a huge amount of immunology and developed the virology in parallel. All of these things could be important. And then, that's the antibody side. What do we learn on the cell side? Well, we went after a cell-based vaccine, but the cellular immune responses generated in the Merck vaccination didn't appear to protect. And so cell-based vaccines fell out of favor. And what do we get next? We have a really fascinating thing with the CMV-based vaccines that don't generate neutralizing antibody, but appear to not necessarily prevent infection, but to control infection in monkeys. And so those vaccines, the CMV-based vaccines, need to move forward for testing in humans. And that's one of the additional new directions that is not antibody necessarily mediated. I think the final part of this is animal studies. So, we got really excited by the cell-based immune generating vaccines because of their impact in monkeys against what we called infection, which was an intravenous challenge with a relatively easy SIV, HIV chimeric virus. Monkey models have not been that helpful so far, and if anything, they may have misled us early on. So developing correct animal models, I think, is really important. And this was a lesson learned and used in COVID. We now know that the animal models did predict human protection, and that's a very important thing to note. I think that the very successful response to vaccine development for COVID-19 is really tied in very closely to the ability of the scientific community to answer questions around HIV, whether it is neutralizing antibody specialists in HIV who are at the forefront of developing the correct assays or standardized assays for COVID-19, or the sequencing groups that really did a huge amount of work, or the development of research capabilities in Africa, which I think are going to serve us well as vaccine testing begins to engage not only high-income countries, but low and middle-income countries. And it gets to the idea that we can build sustainable research and development capability throughout the world on a platform that was initially developed around HIV and the immense scientific questions that had to be answered and still remain unanswered for HIV.
So, Susan and Jontraye, Dr. Kim started by making the point that an HIV vaccine has been more difficult to develop than the COVID-19 vaccine was because it's a different virus. Do you want to start by giving us your thoughts on that?
So the success of the COVID vaccine is really remarkable and very exciting. But people do ask, why is it taking so long to find an HIV vaccine if it was so easy to find a COVID vaccine? And I would say that, first of all, the COVID vaccine work was built on the structure of many decades of work on HIV vaccines. The actual structure of the vaccines that have been developed, some of them were developed specifically for HIV, but also COVID is a really different virus than HIV is. So HIV is what I would call a kind of wily virus in that it attacks the very immune system that's supposed to control it. And when you become infected with HIV, you get infected for life. The virus inserts itself into your own DNA or genetic code and sets up a lifelong infection. And so that really causes difficulty in once you become infected, people don't spontaneously clear the virus. Whereas with COVID, once you become infected, you do clear the virus, you get it out of your system, and we know what kind of immune response actually clears the virus. So we had a target for our vaccines. We knew what kind of immune response would be protective. For HIV, we don't have any kind of immune response that we know is actually protective in the natural world in people who become HIV infected. So people don't clear it. And so it's much more difficult. We don't have any goalposts really to shoot for an HIV vaccine, or we didn't until we got some positive results from an earlier trial.
I like the way that you stated that, and thank you for that. So I recognize we have a diverse audience with folks who do not possess a highly scientific background. So I just like to regurgitate something that you said to ensure that our audiences can follow. So HIV and SARS-CoV-2, which is the disease that causes COVID-19, those are two different viruses. And so you're saying that HIV embeds itself into our DNA, therefore making it a lifelong or a chronic illness, while SARS-CoV-2 does not and doesn't present itself as a chronic illness. And our bodies have the ability to naturally heal and destroy the viruses that cause COVID-19 disease. Is that correct? Did I understand that correctly?
You did. You did. That was beautifully stated. So we do clear the virus and get rid of SARS-CoV-2, the virus that causes COVID. And that's how people end up recovering from COVID. Now, we don't completely understand the long haulers and what's actually happening there, but we do think people have cleared the virus from their system. So it doesn't set up a lifelong infection in the way that HIV does. That people make what we call neutralizing antibodies against SARS-CoV-2. And then after they make those neutralizing antibodies, they clear the virus infection from their bodies. With HIV, the virus mutates so rapidly that people do develop neutralizing antibodies against HIV, but they're always 2 steps behind. So by the time they create neutralizing antibodies against one configuration of HIV, it's already mutated into another configuration of HIV. So then the body makes yet another kind of neutralizing antibody, but the virus is always 2 steps ahead. We do, though, have these things called broadly neutralizing antibodies that appear to neutralize a wide array of HIV viral particles. And that is a success that we've had recently in HIV vaccine development, is in using these broadly neutralizing antibodies like the VRC01, which is the name of one of the kinds of neutralizing antibodies. And it did provide some protection against viruses that were easily neutralized by that virus. So it was a proof of concept in human trials that, in fact, you could provide protection against becoming infected with HIV if you have the right broadly neutralizing antibodies on board.
Yeah, just a little backstory for that. So I know a lot of people are probably wondering, what is VCR01 and where did the concept originate from? So this broadly neutralizing antibody was discovered in a person living with HIV for many years, and the person was not receiving treatment. And I think it was approximately 15 years or so after they had acquired HIV is when they realized that this antibody was present and played a critical role in fighting off the infection in this person, which then resulted in replicating the BNAB to be used in clinical trials after that.
And I think it's one of the big successes of the HIV vaccine field that it proved this concept that broadly neutralizing antibodies can actually provide protection against HIV. It was a modest level of protection, and it was only against the viruses that were sensitive to the VRC01. And so what we know now is that we need a cocktail of broadly neutralizing antibodies in the same way that we use a cocktail of drugs against HIV for treatment. We need a cocktail of broadly neutralizing antibodies probably in order to provide protection against HIV. And so that is one of the strategies that's currently being evaluated that may move into efficacy trials.
Yeah, I really appreciate how you mentioned that, the study was a proof of concept versus efficacies. It gave us kind of like, the ingredients and information that we need to continue forward with the development of a vaccine or some other modality that could be long-term, if you will.
And so there are really some great developments in broadly neutralizing antibodies that are being currently evaluated. Another strategy for vaccines is to actually try to help the body to create its own broadly neutralizing antibodies. And that's been a very tricky area for us to pursue. Some of the earliest vaccine efficacy trials that were done were hoping to create broadly neutralizing antibodies or neutralizing antibodies against the virus, but those were not successful in providing protection.
Do you have any guidance or any recommendations for, people who are really wondering what's the difference and then why should I even consider either option?
One of the reasons that we think about active vaccination being really important, in addition to studying broadly neutralizing antibodies, is that the neutralizing antibodies will go away on their own. And what we would hope for from a vaccine is that you would continue to hold on to these memory cells so that if at a point in the distant future you were exposed, that you would actually be provided protection against HIV. So we'd hope for something like some of the vaccines that you only get once in life that you don't need to get booster doses for, or something like tetanus shots where you need to get a booster every 10 years, that would be something that we would be hopeful that we could develop rather than needing to get something every 6 months or every year. So one of the advantages to an active vaccination is that in an ideal world, you'd give it to babies so that their immune system is trained so that if when they grow up, they become exposed to HIV, they're actually protected in a lifelong way from HIV infection. That would be the ultimate goal of an HIV vaccine.
Okay, thank you for that.
And so I think one of the other questions that gets asked is, where are we 40 years into the epidemic? Why don't we have an HIV vaccine? And what have we learned from these vaccine studies? Have we really learned anything? And we've learned actually a tremendous amount from each of these human clinical trials that we've done. There are any number of lessons that we've learned from those studies. And I'm happy to sort of talk through what some of those lessons have been. But I think it's really important to know that even when a trial, a large-scale efficacy trial or proof of concept study is not successful in providing protection against HIV, we still learn an enormous amount from those trials. And Thomas Edison was supposed to have said, when he was asked why he had 10,000 failures in trying to create the incandescent light bulb, he said, I didn't have 10,000 failures. I didn't have one failure. I just proved 10,000 ways that this wouldn't work. And once I've eliminated the ways that it won't work, I'll find the way that it does work. And of course, he did. And so we don't think we're going to need 10,000 efficacy studies to get to protection. We're building on the lessons that we've learned from each of these efficacy trials to make the next large-scale efficacy trial better. But all of these efficacy trials test, is the vaccine safe? And we build a huge portfolio of information about the safety, first in animals and then in humans in small numbers and then larger numbers, to be sure that the vaccines are safe. We look at what the immune response is like, but the efficacy trials also look to see, do they actually provide protection against infection? One of the other things was the RV144 trial, and I think that's a really important vaccine trial to highlight because it's really our one active vaccine trial that showed some level of protection. There was a 60% lower rate of infection in the people who got the vaccine compared to the people who got placebo or just a blank injection at 1 year after the vaccination. And then by the end of the trial, it was only a 31% reduction in the risk of HIV infection. So not a slam dunk in the way that the COVID vaccines have been, but enough to sort of give us a sense that, gee, if we had as strong an immune response as we saw at 1 year, maybe we could actually build on that and make it even better. Remember, I said earlier that we don't have a goalpost for what it is we're shooting for in an immune response because people don't clear the virus themselves. What that gave us was what we call a correlate, or an immune response that appeared to correlate with protection against infection. And so the goal there now has been, can we create an immune response like we saw in the RV144 study, but stronger than that, that would provide protection in future studies? Now, we tried doing that in another study that took place in South Africa, and unfortunately, it was not successful. And there are a number of reasons why it may not have been successful, including that the risk of infection was many-fold higher in South Africa than it was in Thailand. So if you take what's a weakly effective vaccine, it may just have been overwhelmed by the high risk of infection in South Africa.
So, Dr. Buchbinder, for those two trials, do you feel like the two populations were different? And I'm also sure that the social determinants of health were also different across both populations. So do you think that that may have contributed also to the differences in the results?
Yeah, the thing is, there's so many differences that happened in that trial. So they created a new vaccine because it needed to be geared towards the kind of viruses that are circulating in South Africa rather than the ones that are circulating in Thailand. They gave it with a new adjuvant, which is a kind of chemical that boosts up the immune response to the vaccine. As you said, there are differences in the populations themselves, in the rate of sexually transmitted infections, in the kinds of bacteria that you find in the genital tract that might make you more susceptible to HIV infection. And then all of those social determinants of health that may increase your risk of becoming HIV infected in one population compared with another population. So the viruses were different, the populations were different, and the vaccines were different. And so what we're trying to do now is tease out which of those differences might have accounted for the fact that we did not see protection in the South African trial that we had seen earlier in the Thai trial.
Okay, that makes a lot of sense. So ultimately, we should remove the word fail from our vocabulary and say that the last 40 years of HIV prevention and treatment research have really just, gave us additional information that we needed to expand the toolbox, because we have come so far compared to the early 80s. And here we are now. We have so many options that we didn't have 3 decades ago. So we should really remove the word fail from our vocabulary when talking about HIV prevention and treatment research.
Right. I think, what would be failed is if you set out to answer a question and did the study poorly, or it was designed poorly, and so you didn't get an answer to your question. Science is really about doing an experiment where you don't know what the answer is going to be, and you've got some hypotheses, and you're testing those hypotheses to see whether or not they pan out. These studies have been successful in that they've been extremely well conducted, and they've gotten us often quick answers to important questions that we have, and they've helped shape what our hypotheses are about what it is that's needed.
I completely agree, and I think you have both made excellent points to shift this discussion from why the HIV vaccine development effort has, quote, failed, to instead focusing on what critical information has been learned along the way. Is there anything else either of you would like to share about key takeaways from the last 40 years of HIV vaccine research?
Can I just shift gears for a second and to just learn more from you about what your opinion is of community engagement in all of this? Community engagement as well as good participatory practices with HIV clinical research and trials, but also with COVID-19. In response to that, I know that many of the same community engagement strategies that have been used in HIV prevention research has also been carried over into COVID-19 research. So I'd just like to learn more from you, your thoughts about effective community engagement during all of this.
Yeah, I would say that the whole field of community engagement has been changed forever by the HIV prevention and treatment advocates throughout the epidemic. Before HIV, I would say that there was fairly minimal community engagement in the research that was being done, and particularly for biomedical trials. And the good participatory practices, which is this template of how it is that you should engage communities in your research, actually grew directly out of the HIV vaccine effort. And so it's really served as a roadmap for the best ways to engage community in the research that's being done. And that's, I think, really a critical part of success. If you're coming from the community, if there's a research site that nearby that does HIV vaccine research, all of these sites have community advisory boards. And if you can join the community advisory board, you get mentorship from peers in the community advisory board, as well as from staff members from that HIV vaccine trial site that can help you both learn the science of HIV vaccines, as well as learn how to read protocols and how to advocate for the kinds of input that you think is needed in the research itself.
And also to everyone else, I encourage people to also advocate with politicians and funders to keep the field moving. And religious leaders in your community as well.
Those are great points. Thank you for raising those. And we really do need that kind of advocacy because we need the research to continue. There was tremendous advocacy around COVID vaccines, and COVID got a lot of resources devoted to it. We need to be sure that people understand that there's still a very severe HIV epidemic globally. There are 1.7 million new infections each year and 700,000 people dying of HIV each year globally. And so we really need to address both prevention and treatment, and vaccines is a critical piece of that. Another topic that we touched on earlier but didn't completely get to is the ways in which HIV vaccine research really supported COVID vaccine research. There are many, many different scientific fields. The COVID vaccine effort really built upon lessons learned from those. As I mentioned, some of the COVID vaccines themselves were built on templates of the kind of vaccine that's been developed for HIV, that's being tested for HIV. We standardized laboratory studies so that we can understand in really clear ways across laboratories what it is we're learning from these trials. And we hone the statistical approaches that are used to try to give us as much information from the trials as possible. So those are really important contributions. And then the COVID vaccine studies were actually conducted at HIV vaccine and HIV prevention and HIV treatment trial sites. That's the way that we were able to launch these tens of thousands of person trials so quickly, was that we already had staff and investigators throughout the world who were experienced in running these kinds of clinical trials. And without the HIV vaccine effort, I think it would have been a much more difficult course to have followed.
Right. So not only has the HIV vaccine field contributed to scientific advancements that helped develop a COVID-19 vaccine, but also was instrumental in community engagement and providing the infrastructure necessary for large-scale vaccine trials. Well, that's all we have time for today. Thank you again, Susan and Jontraye, for your contributions to the field and for participating in this discussion. And thanks to our listeners for joining us. As a reminder, click on the links in the show notes to listen to the HIV Unmuted podcast series produced by IAS, and to join us for the third podcast in this series, which will address how best to increase advocacy and investment for an HIV vaccine to continue moving the field forward.